introduction of Professor Goel. Since the year 2007, he is working as a professor. He is working at BU since October 2007, but as a professor since 2013 in the Department of Botany, University of Delhi. He did his PhD in 1999 from the department itself, but then went on to do some postdoctoral fellowships, first at the Department of Horticulture, University of Georgia, US, to 2005. Then he moved to France and for another postdoctoral fellowship at IRD, which is Institute, I hope I pronounce it right because this is in French. This is Institut de Recherche pour la Développement in France, and his tenure was from 2005 to 2007. Then in 2007, India called him back and he came back to India. He joined as a reader in Northeastern Hill University, Shillong, and he served for a few months only, like from June 2007 to October 2007. And then in 2007, he joined Department of Botany, University of Delhi. He has also availed another research fellowship which is at Kihara Institute for Biological Research, and this was when he was still doing a PhD in the lab. He is now on the editorial boards of many journals, and he's academic editor for PLOS ONE, he's guest editor for MDPI Horticulture, and he is on the editorial list of many other journals of repute like BMC Genomics, Plant Journal, Annals of Botany, Heredity, Frontiers in Plant Science. The list is long. He is also a member of many prestigious bodies. He is an elected fellow of Linnean Society of London, UK. He is a member, American Association for the Advancement of Science, Association for Promotion of DNA Fingerprinting and Other DNA Technologies at CCMB Hyderabad, Society for Phytomorphology, University of Delhi, and Indian Society of Translational Research at JNU. He is also member of various selection boards. He is part of various boards such as Board of Studies, Jamia Millia Islamia, NIPGR, DTU, Sikkim University, NEHU, and others. He is also EC member of EdNet, CCMB, Hyderabad, and a member of Academic Council, Jawaharlal Nehru University, Delhi. He has very impressive list of publications. He has more than 65 publications published in International Journal of Reputes, 7 book chapters, 13 invited lectures. He has edited a book, and he has more than 60 abstracts in various conferences. As far as the research grants are concerned, he has already completed 11 projects, and with very high amount of grants from DBT, UGC, DRDO, DST, etc. He has 4 ongoing projects as PI and co-PI, and there are many research scholars working in the lab already. His list of achievements is long. We are already little delayed, so with these few words of his formal introduction, now I would invite Professor Shailendra Goel to take the dais and conduct his lecture. Over to you, Professor Goel.
Good afternoon. Thanks for inviting me here. Special thanks to Professor Rani. I think I know most of the teachers in this department, so this is actually kind of nostalgic for me. Many of them were doing PhD when I was doing PhD, so I'm feeling good, actually very good. So thanks for inviting me here. And the talk is just an excuse because I was forced. Anyway, we just can interact. There are slides to run. I can run them in a minute, doesn't matter. It should be more, I think, a two-way talk. So you are free to interrupt me, ask questions. I'm not trying to showcase my research at all, but I always loved genetics. So when I came out of MSc, I'm a small town guy. I came from a very small town called Muzaffarnagar, and I came to Delhi University for PhD only, where I met Professor Vijay. I guess you have a very good department and very good people to guide you. So maybe my guidance is going to be redundant. So what we will do, we will just talk about what is happening around the world, what is happening with me, what is happening with you. So one thing which I noticed over this time is I couldn't detach myself from genetics. So you can think I worked in Department of Horticulture, I was still doing genetics. I came back to botany, I'm still doing genetics. I don't do anything else except genetics. But the tool kept on changing over the years. Whatever I was doing has to be reinvented in 3 years. So during my period of almost, I think, 30 years now, I think 25 years. 25 years I changed technologies like crazy. Every 3 years I was learning something new. And right now I am learning NGS. So because of that, I realized that this is evolving so fast and the speed of evolution has increased because of computers. And now you are going to get AI. So I think it will be crazy. Now we won't be able to catch up even. So I think you guys who have to catch up because we are getting slow. And AI is going to bring us to the speed where only tech-savvy people can handle it. And the statistics, we used to know mean, median, mode, X, chi-square test and t-test, it's all gone. Nobody does that anymore. Now you have multi-layered AI-based multi-layered analysis. There is vector machine. Things are crazy. So I was tech savvy in my time, but now I don't feel like that anymore. I feel like redundant. Students who come to my lab are more tech savvy. So whenever I am stuck, I will call them. "Bhai, ye kya naya aa gaya? Bata do mujhe." We kept pace till say Facebook, and then it went crazy. Twitter आ गया, फिर क्या Instagram आ गया. I just lost track. So I stopped doing all that. So your generation, I think, will be able to do a better speed than us. And I think it's with every generation. Before us, people were taking more time and we were thinking क्या कर रहे हैं हम लोग स्पीड ले लेते हैं और अब हमें लगता है कि अच्छा वो ये कर रहे थे अब तुम भी यही करने वाले हो दूसरी जनरेशन आएगी वो स्पीड लेगी बट I think that's how you pass on the torch. So be ready for that. And this is not going to be research. It is going to be only telling you how it is moving forward. Okay. So evolution of genetics. The idea is that we need everything fast. That's I think is the goal always. Science goal is also to speed up, and people have been doing that. So we will walk through markers, which you know already. I am not going to teach you about markers. I am just going to show कि किस तरह से marker ने speed ले ली. Then I will be showing you a bit about breeding, how breeding is speeding up because breeding is a major bottleneck in genetics. Another thing which I noticed over the time is that people talk about genetics, they talk about Mendelian genetics, and they just ignore population genetics, which is very important. Many people do work on population genetics, but they don't even themselves understand what they are doing. So some tidbits here and there. You feel anywhere you want me to explain it more, I will do that. So these are some of the plants I work on. And if I put more figures, it will be very difficult to explain. I work on all kind of plants because I collaborate a lot. So many people collaborate with me. My list of collaboration is like 50, 60 people. And because of that, but these are some of the programs which I developed. So first is Nymphia and Hippophae. So Nymphia and Hippophae are some economically important plants, but they are not crops. And the idea of crop is it is domesticated, being grown in a crop environment which has a different ecology. The idea of crop growing is to give it everything to maximize the output. So when a farmer goes to field and he plants anything, his idea is okay, I will give it everything, all the fertilizers possible or everything. Idea is within 3 months I should get my crop and it should be enough to sell it so that my input is lesser than the output. So these crops which we are identifying now are actually not domesticated. The domesticated crops are those which started at the time of say when human being started from gathering to grower. It became an agriculture based person. So you have crops like Vigna and Carthamus which are from that time. Over the time human population grew, so the crop genetics moved towards 3 major crops which are cereals, rice, wheat and maize. But we ignored everything else. Why? Because demand was less. People were more towards alleviating the hunger, trying to get everything enough so that people can eat. But then there are plants also like primula, which doesn't have any economic importance, but ecologically they are very sensitive. They have a special niche in the environment. They grow at only high altitude. So I start working on that also. And that's why I'm dealing with both crop genetics and population genetics. So in plants, you can divide genetic research into two parts. One is population genetics, another is biparental genetics. Now, don't take it in a textbook manner. I'm trying to divide genetics in a way that it reaches you. Now, the goal of population genetics is to understand the genetic diversity, which often lead to policies for conservation. Conservation of plants, conservation of environments. Then you have also evolution and species relationship, which we also study based on genetics. And you must be hearing about it. The new AGP, the one which we follow, is actually based on the evolutionary relationships. And biparental genetics is mostly crop genetics. You can do it with any plant, but it has contributed a lot to crop genetics. So in biparental genetics, you have goals like linkage map or identification of genes which you want to, say, transfer to some other plant. Then we also work on germplasm characterization, which is actually base for any crop improvement program. You first pick up the germplasm, you characterize it, figure out all the traits which are agronomically important, and try to start your breeding programs for bringing these traits together. Now, breeding changes from all over the world. I have quite a few friends who are working on companies like Monsanto or Pioneer, which is now everything is Monsanto actually, they bought up everything. But there were companies like Seminis, also bought by Monsanto, which used to focus on vegetable plants. And the breeding for each region is different. So if you look at India, what we eat in cucumbers is different from what they eat in Mexico. They want the cucumbers as pickled. So they are smaller in size, easy to eat, and they should behave well when you try to pickle them in acetic acid, vinegar. So the breeding programs also change from the world, from one place to another place. When people start working on that, the then idea is to grow or collect all the germplasm and look at it from region wise. In each region, plant is acclimatized to that particular environment. If I try to develop a crop which is growing, say, in northern part of India, and I try to transfer it to southern part of India, it might not work very well. So regional distribution of traits, regional distribution of demand of the consumer, they all are taken care when you start with the breeding programs. So the genetics is normally introduced as this, that you cross two parents, you go to F1, in F1 the combination happens, in F2 you map it. And that mapping is based on markers. And normally it gives you something like this. Now this is from a paper in 1990s, this Barley linkage map. And this was the time when I joined my PhD. And by the time I did my PhD, figure changed to this. This is what we call as high density maps. The idea is to increase the number of markers. So we will come to that, why. But science never satisfies. The next goal was this. What is this? You're trying to figure out the whole genome. You're trying to assemble the genome. And your genome assembly includes the genetic map, physical map, and even the sequence-based map. And you want to bring all this information into one place. By the time I went to my first postdoc, figure started looking like this. What is that? This is a genome browser. What you are trying to do is, you are trying to bring all the information together in one place. Now on the top is a chromosome. And you see a red spot on that chromosome is actually a sliding window. That window can be taken over from one place to another, anywhere on the chromosome. What you get below is the information contained in that particular window. And that information includes the repeat sequences, markers which have been identified, genes which have been identified. Your idea is that I don't work at all. I get everything from there, which is very easy for getting a PhD. But you know, it becomes more difficult to do a PhD if everything is there already. Because you need to find a new problem for doing a PhD. So this is 2000, say 3, 4, and genome browser was a new thing, HAP thing. People used to brag about it. Oh, I created a genome browser. But now it is so common that nobody cares. So after that, we go back to maybe tracing the history, and you remember all the markers. Actually, I'm missing a marker here. The first marker, molecular marker, came from Lithuanian and Habi. If you recall, they are allozymes. So allozymes were being done on gel plates. They will stain them, trying to figure out. And if you did 4 allozymes in your thesis, you are a king. You will be given a very good postdoc. Maybe you will get a paper in Nature. But that was in 1966. Time moved on. We moved on to markers like RFLP. RFLP is, say, roughly time of 70s. In that duration, the way of detecting the band was radiography. So you do a labeling P32 probe and you expose it. 1 week down the line, you get 2 markers. So at that time, people started creating small genetic maps, but they were not happy because time duration was too long. So 1990s, Mulis came, you got PCR. So people shifted to a PCR. Okay, this will give us a speed, more marker. So one very good marker which came out of PCR and used for a long time was rapid. Now rapid used to give you 5 to 6 marker in a day, say about 3, 4 hours, 5 hours PCR. And in that period, you get 7 markers. So for a while, rapid lingered on, but people were not happy. So then they combined RFLP with rapid, and came out AFLP. Now AFLP was giving you 40 to 50 markers, and doing AFLP again was radioactive based. So you have to expose it for a week, and after a week you at least got 50 markers. So your PhD duration from 10 years came down to 5 years. So then it keep on changing, but people then got stuck on microsatellites for a while. The reason is all the other markers on the top except RFLP are actually dominant markers. For a geneticist, dominant marker is a bad news. You want a codominant marker. You want to have all the information about alleles. Genetics is all about alleles. If you can think in terms of alleles, genetics is easy. Everything else is just a tool. It changes, which is what I'm trying to show. Molecular biology is a tool. You are getting markers, but eventually you need to know how to use them, which is genetics. So कोई molecular biologist है तो please offend नहीं होना, मैं geneticist हूँ. So मैं तो वही sell करूँगा, ठीक है? So this brings up to these fundamental questions about markers. What is a molecular marker? What is a good marker? Why we need so many markers? Which one to use? And where are markers heading to? So if we go with the first question, what is a marker? Marker is anything which indicates to the location. Now you started from your home, you are going to a friend's place, you will call him कि मैं first time आ रही हूँ या आ रहा हूँ, so कहाँ है? वो क्या कहेगा कि वो आप आ जाओ से close to Ames, वहाँ से left मुड़ जाना, वहाँ कहीं एक पीपल का ट्री है, उसके सामने मेरा घर, right? That's a marker. Same thing a molecular marker is doing for you. It's just molecular, but it is actually indicating to you where in the genome you have to look for it. So genome is a landscape. Now if you want for whole Delhi one marker, that's not going to work. Same is for DNA. It is not going to have 1 or 2 markers. You need thousands of markers so that each location, each loci, wherever it is in the genome is marked and you can reach to it. That's the whole idea of genetics. So you are looking for alleles, and those alleles, what you are trying to look for is the allele of your choice, the one which will give you benefit. So this is the idea about genetics in the first place and in the last place. From the beginning to the end, this is what genetics is. So let's go. So this is what we are looking for, right? That's the marker. What is a good marker? It should be easy to handle, it should be cost effective, and it should be reliable. Now you started from your home, your friend told me कि वो पीपल का ट्री है उसके सामने आ जाना. The first question you बोला कौन सा पीपल का ट्री, मेरे घर से निकलते ही 10 पीपल के ट्री हैं. So what you need is a location, the precise, reliable location which doesn't confuse you. Now you will ask, does it apply to genome also? It does. genomes have satellite DNA. You know what is satellite DNA? And no question is stupid. मैं अभी भी इस स्लाइड को बैठूंगा ना देखने, मुझे इसमें से मिस्टेक मिल जाएंगी. वो होता है, वो पार्ट ऑफ लाइफ है. डूइंग ए मिस्टेक इज पार्ट ऑफ लर्निंग. सो बोलना शुरू करो. So satellite term is used at multiple places. The satellite chromosome also is there. You have a unit extra constriction attached to the chromosome where the nucleolus is attached. But that's besides the point. The satellite for DNA came from the extraction protocols. In 70s, the DNA extraction used to be with cesium chloride gradients. So when you do cesium chloride gradient, you used to get a major band and a minor band. So anything which is above the main, like Earth, above it, Moon, is a satellite. So they called that as, at that point, satellite DNA. Later on they figured out that it's all repetitive DNA. And that's why the satellite DNA is still being used loosely for any kind of repeat sequences. So अब satellite DNA का यदि आप मार्कर बनाने की कोशिश करेंगे, it will be not a reliable marker because it is present in multiple places in the genome. What you need is a unique marker for each unique location. So we try to find those markers which will show you specific allele to the loci and then try to work on that. So why do we need so many markers? I already told you, if you are dealing with Delhi, you need enough markers to cover Delhi. And the same thing you are doing in the genome. Your genome landscape has to be covered enough. That's why we need more and more markers. Now which one to use? Now that depends on the question. What kind of problem you are facing. So if you are working on diversity, you will choose markers which are known to work well for diversity. If you are working on mapping, then you will choose accordingly. If you are doing some kind of pedigree analysis, then you will choose accordingly. So your marker choice is based on the kind of problem you undertake. Where are they heading to? From the beginning, they always craved for more resolution, faster speed, more number. That's the whole idea. So the idea here is resolution power, number, codominant marker as preferred, and then speed. This is the base of whole genetics. And in this era of genomics, we are dealing with SNP. SNP is a codominant marker and it can come from different portion of the genome. You can choose from DNA, you can choose from transcriptome, you can choose from something else. So you can design your SNP to your choice and use it. So first question you have to ask, what I am targeting in the genome, and then accordingly pick up the data set from which you will identify the SNPs. But SNP has one bottleneck still. You need bioinformatics support. So you need to have enough bioinformatic information, but this is slowly fading away because bioinformatics is, in my time, bioinformatics means blast karna. And you all know blast kitna aasan hai aajkal. So there will be time, maybe in next 3-4 years only, where you don't need to do much in bioinformatics. There will be pipeline which will be user friendly, one click and you get your information. So that bottleneck will go away in a few years. But the cost is the major reason right now. So next set of slide is trying to introduce you to cost, how it came down. As I said, I'm not trying to show you research, I'm just trying to show you the journey of genetics, how it went on. So these are some timelines, you remember them? I think आती भी होंगी syllabus में, आप लोग पढ़ते होंगे इसके बारे में. So the first one is maximum Gilbert and Sanger sequencing method, which is in 1977. Now next major change came in 1986, almost a decade. What did it change? It changed to a chromophore based system. So you don't need to do autoradiography anymore, which speeded it up. I will show you a pictorial later which will explain it better to you. Another 10 years, you have capillary based DNA sequencer. Another 10 years, you have now NGS. Another 10 years, things kept on changing. So every 10 years, there is a major change. And this period of 10 years is now reducing because now we have computers and AI coming up. So I personally have been upgrading myself every 4 years, that 5 years initially. Now I am upgrading myself every 3 years, and it's getting crazy. Now it's coming to 2 years, maybe soon 1 year. So it's changing very fast. Maximum Gilbert you all know. what it is, so I won't explain. We skip it, right? Move on. Sanger, one guy who got 2 Nobel Prize. बहुत कम ऐसे हैं जिन्हें नोबेल प्राइज 2 बार मिला है. कितने हैं? कोई और गिना सकते हो? हाँ? There are 5 in total history. 5 people. And Marie Curie के लिए Curie effect आता है. पता है क्यों? Marie Curie is also known for Curie effect. Marie Curie was not the only person who got Nobel Prize. Her whole family was Nobel Prize. Her husband was also having Nobel Prize. Her daughter, I think. Yeah. So that family is known for Nobel Prizes. Something special in their genetics. So.
Likewise, Krebs, linkage group, four Nobel Prizes.
Linkage group.
Muller and all.
हाँ, Muller.
Morgan, Muller and Studwent. They also got many Nobel Prizes.
हाँ, नहीं, they got 1.
ना.
5 ही हैं टोटल जिनको डबल है, और बायो में सिर्फ
फैमिली वाला होगा ना दिस ग्रुप.
हाँ, बायो में सेंगर ही है. सेंगर इज द ओनली वन जिसको 2 बार है. एक वो पॉलिंग है, उसे है, उसने लेकिन विटामिन सी भी किया था और पॉलिंग एक्सक्लूजन प्रिंसिपल भी दिया था, तो फिजिक्स में भी मिला है और बायो में भी मिला है. ऐसे 3-4 लोग ही हैं. एनीवे, सो डीएनए पॉलीमरेज आपको पता ही है ये क्या करता है. एंड यू डू होल थिंग, इवेंचुअली यू गेट दिस. व्हाट इट रिक्वायर्स? Whole reaction, running the gel, doing autoradiography. You spend like 2 weeks. I did it in my PhD. She used to come back. मेरी पिक्चर यदि अच्छी हो जाए तो she used to comment, आज तो बड़े खुश नजर आ रहे हो. So, you know, but it was painful, really painful. So that's the first thing where I moved on to new technology. So from this, the Kihara Institute Fellowship, Japan, I did something else rather than radiography, and whatever I was trying to do in my lab for 2 years, I did there in 3 months. So that's how the science impacts from the new technology. And this is what changed it. Fluorophores and lasers. This slide explains its pattern. Initially it was 1 fluorophore, so you still need 4 lanes. Then people thought, why 4 fluorophores? Why can't 1 fluorophore, we can have 4 fluorophores. If they had 4 fluorophore, the whole thing from 4 lanes shifted to 1 lane. Now then people started saying, why gel? We will shift to matrix, some capillaries. So whole thing shifted to capillary. Initial sequencer were 1 capillary. Then they became 8 capillary, then they became 16 capillary, then they became 96 capillary, and now it is, I think, 384 capillaries. So, you know, speed kept on increasing. And what it did, it brought to you this everybody knows, and Human Genome Project, because it made possible the Human Genome Project. Now, in Human Genome Project, 2 points only I will like to point out. The cost was $3 billion for 1 human genome. And it took 13 years. And at that point, a huge conference was called. You remember, maybe you don't remember. आप लोग शायद पता नहीं, पैदा भी नहीं हुए. So there were 2 people. Watson had a separate group and Venter had a separate group. They were fighting against each other to come out with the first Human Genome Project. And their strategies were different, but that's a huge explanation, so I'm not going into that, although slides हैं नीचे इसमें. So this 13 years was a huge fanfare कि हमने 2 साल जल्दी खत्म कर दिया. So after this is the a lab doing sequencing at that point. So many PCRs, so many sequencers, so many servers. अभी आपके कंप्यूटर पे शायद उतनी, जो आपका लैपटॉप है वो उतनी पावर रखता है कि वो इससे सबको असेंबल कर देगा, जो उस टाइम इतने बड़े बड़े सर्वर्स में था, जो ह्यूज रूम फिल्ड होते थे. सो ऑल दिस चेंज्ड व्हेन न्यू एज सीक्वेंसिंग आई. सो द NGS changed the scenario from all that to a small room. Now you can do all this in one small room, and the impact we will see in downstream. This is Moore's law. Moore's law was actually proposed for chips, computer chips. On computer chips you have transmitters, radio transmitters. So Moore, at that point, he just made a simple observation that every 2 years the number of transistor on the chip is doubling. That's what Moore's law is. So that became a rule in economics and people started applying Moore's law everywhere, so they applied here also. And you can see that it did follow for a while and then it crashed. Cost is so low right now that you won't believe it. So I go to previous one. 2006, Illumina, they proposed that we can do now human genome in $300,000, which is compared to 3 billion. So you imagine how much cost has fallen. 10 years later, 2004 or 8 years later, it came down to $1,000. And in 17, it was already $100. So the cost is falling. And that's the reason now you are seeing personalized medicine. You can get your genome sequenced. They will analyze it and tell you, okay, you have these genes, so you are most likely going to have cholesterol problem. So you start early in your life. Although many people oppose it also, I also oppose it. कि और मेरा खाना बंद हो जाएगा, मैं सारा टाइम टेंस रहूँगा कि मेरा कोलेस्ट्रॉल बढ़ रहा है, तो कोलेस्ट्रॉल तो बच जाएगा, मुझे हाइपरटेंशन हो जाएगी. So, but personalized medicine is now common, and at the time of say COVID, many people followed it. There were companies, if you have COVID and you are starting to get some complication, they will sequence your genome and tell you what kind of medicine will be better for you. So personalized medicine is now becoming a possibility just because of that. And cost is just $100. So even one of my student, she was in US, and in 17 only she told me that she is just coming out after giving her DNA for sequencing. I was like, why will you do that? So she said they were giving it to me for free. All they want that, okay, my DNA sequencing they will keep for analysis. So she did that. She was fine with it. And she got a final analysis and they told her you are 2% Viking. So I was like, okay, now you buy those pointy heads because you are a Viking. So, but you know, things are moving very fast. And genome sequencing first time looks like a Herculean task. Now it's easy. In my whole PhD, I generated 24 KB data. And now the student who comes to my lab first day, within a week, he is generating TB's data. So there is no comparison. Things have changed totally. This bioinformatics is now more of a requirement because data generation is easy. The problem is making sense of that data now. So anyway, so you saw this part. Overall, why this happened? Because there are several things which came together. It's not only one thing. It's physics, chemistry, biology, everything came together and made it possible. So that's why science has to be interdisciplinary, cannot be with one thing. So I am IQAC, which have been told at many points. So one part of my job is to go to different departments. And whenever I go to these departments, especially physics, chemistry, they are very ugly. They always tell me, आप लोगों के साइंस में सारा काम तो हमने किया है, आप देख लो, जो भी हुआ है वो सब हमने किया है. मैंने कहा आप क्या कर रहे हो, खाना खा रहे हो ना? कहते हैं हाँ. मैंने किया है. तो उसके बाद वो चुप हो गए. उसके बाद से उन्होंने वो आर्ग्यूमेंट बंद कर दिया मेरे साथ. मतलब they were friendly actually. So we used to talk about it. So you have general progress in technology, significant reduction in cost. Reference means more and more genome are now available. People know what you are expecting within the genome when you sequence it. And then broad range of important biological phenomena, which we try to analyze and figure out from this data. This is traditional genome. You figure out back clones, you find a tiling path. This is what I did in my postdoc. Horrible. And then, now everything changed. No E. coli. I used to handle a library of 1 lakh clones, but nobody does that anymore. No need to replicate 1 lakh clones, nothing has to be done. Everything is on beads and it's done easily. At that point, labs used to have hundreds of minus cities, features, because libraries has to be stored. So Rodwing is known for back libraries, and Rodwing's lab has a huge basement filled with something like 200 minus cities, all having different back clones from different systems. All this changed. So we switch back to now markers. You now know why markers changed. And these markers now we are talking about is microsatellite and SMP. One thing you have to remember that they both are codominant markers. But that's the definition of codominant and dominant. If I talk about rapid, how will you explain it's a dominant marker? So definition is still stays same, whether it is showing you both alleles or not. And rapid either amplifies or doesn't amplify. So you're not seeing the other band, and that's why it's called as a dominant marker, which is not the problem with microsatellites. It amplifies both the alleles, and alleles are showing difference in their size. So these two markers are a little bit which I will talk about. Then we will shift to breeding. Microsatellite markers everybody knows, so I won't explain. We move on. But it used to be a tedious job. The first student who worked with me, the one who sequenced her genome also, she did this. And there, what you have to do is first digest your DNA, clone it, pull down it based on some repeat sequences, and then you clone that, and after cloning you sequence it, and in those sequences you figure out which one has the repeat sequences. And it used to be almost a job of say 8 to 9 months. Now, you just sequence it, which is like a day, and you got sequence, you run MESA, and it's so common now, even small labs are doing it. So you get microsatellite. Now the microsatellite is a slow process because at one PCR run you are doing only one marker. But the strength of microsatellite is such that it is going to stay for a while. Any lab who doesn't have any understanding of bioinformatics can pick up the primers for microsatellites and run them, which makes it easy for people working on evolution or genetic diversity. Not everybody is expert in genetics, so what they need is a quick marker solution which they can apply to their system. And because of that, microsatellite is going to stay for a while. You can skip this. This is how you see markers, either monomorphic or you see variation in the size, because microsatellites keep on changing in size because of either unequal crossing over or slippage during the amplification of the DNA. So these are classical gels of microsatellites. You can use them across these species within a genus. If you develop some microsatellites, other closely related species, normally at least some of the microsatellites will amplify there. So this makes it very easy for people working in evolutionary biology or ecology, that okay, we pick up a set of markers, bring it to our system and start working in the nature. GBS is one of the way, as I told you, the SNP can be either GBS, transcriptome based, resequencing based. There are various ways through which you can identify SNP. Like in GBS, which is very common right now, what you do is you digest your DNA, pick up a portion of that, say in 1 kb to 2 kb region, clone it or sequence it, and then what you get from the whole genome, a very small portion is getting sequenced. But your SNPs are distributed throughout the genome, and that's why you get like 5,000, 10,000 markers, which is enough for most of the simple things. This is the protocol for doing SNP. Finally, you get these kind of pictures. Align them, figure out where is the SNP. And eventual goal is to create genetic maps. So this is all about markers. Now you know markers evolved towards higher number, faster information, and of course codominance. Now breeding is another aspect which is normally a bottleneck for any genetic program. So we will just talk about breeding quickly. We are known to biparental genetics. So we have two parents which cross with each other. Now the shift is actually towards from biparental to multiparental. And I have just taken a few examples of that. One is NAM, which we call as nested association mapping panels, or MAGIC, which is multi-parent advanced generation intercrosses. So in nested map, what you do is you pick up a founder population, which means different individuals, and you cross them with the same individual, which I have written here as common parent, and you just keep on crossing them. You will reach to F1. In F2, you will start seeing the recombination, and these pieces will exchange with each other. Eventually, you have a set of RILs, recombinant inbred lines, which have portions from different genomes. Now, how does it help? It helps you to do identification of markers or tagging a gene quickly, because you're not doing it for one plant at a time. You're doing 10, 20 plants at a time. So it speeds up in identification of the genes of choice, marker. Similarly, in magic population, you have a pyramid type of, inverted pyramid type of scheme. What you do is, you create one cross, another cross, then these cross are, the product of these cross are crossed with each other. So you're seeing A to B, got AB, C to D, CD, and then you cross them to get ABCD. So you brought pieces of genome from different portions together. And one argument which you might not understand at this point, but is, it becomes very complicated to track. But it's possible now because you have genomics to support you, thousands of markers, which will be able to identify different genomic portions from different genomes and bring them together. So your whole speed of discovering the important genes has increased. Pyramid breeding, this is an old concept. I think it has been around for more than a decade. So what you are trying to do is bring different traits together in one plant. If you go one trait at a time, it will take years. What you do is P1 crossed with P2, P3 crossed with P4, and then you do selection, choose the gene of your choice, bring it to next, bring it to next. Finally, you got one genotype with all the traits of your choice together. So pyramid breeding is another thing. So these three technologies are mainly about having multiple genomes together and then trying to create populations with them. But one more thing which is picking up, which is not some rocket science, simple logic behind it, is speed breeding. Now what speed breeding does, the idea of speed breeding is to reduce the generational time. If I make a cross today, I have to grow it, wait for the seeds to come out, then I have to self it again and again to reach the S6 population, which means 5 to 6 generations you are waiting. In one season you are taking one, so that means 5 to 6 years it is taking. What they do it now within the growth chambers, and they play around with the light conditions, because you have now very good understanding of how plants flower, how you can manipulate their flowering time. So what they do, they use that information to reduce the time of generation, and within a year, rather than having one, you can have 3 to 9 different generations, 3 to 9 generations within a year. So that speeded it up. For me, in my time, what we used to do is, okay, you have been given this cross, you map it, at the same time create another cross, because by the time you do your PhD, somebody else will come to work on that cross. Now I don't need to do that. I will be doing this within a year, and I can use that cross for my work itself, because within a year I can reach to F6 population, and I try to get mapping in my time duration. Rather than me retiring and somebody else picking it up, I can do it maybe within my retirement time. So this is all in genetics for crops. Markers, which I introduce you, are common even if you do population genetics. I will go through population genetics fast because it is going to take time, and I think you are now saturated. So I will just show you slides. The idea of population genomic is to understand the population structure. You are trying to see how alleles are distributed within a population. So you run markers, you try to get that information and try to see how these are related to each other. Whether there is migration happening from here to there. If you read papers, आता है ना बीच में कि humans were in Africa, then they migrated to Europe. From Europe they spread it to other parts. How do they know it? Based on population genetics. अभी एक पेपर आया है 2 हफ्ते पहले, मुझे नहीं पता आप लोगों ने देखा कि नहीं. It claims that we went through a really bad time, the human beings, and our population shrinked to 1240. It's so low. So all of us are descendants of those 1240. And this happened somewhere in Africa, which might, relate to कलयुग का जो अपहेवल जिसकी हम बात करते रहते हैं इन डिफरेंट अधिकतर जो रिलीजियस टेक्स्ट हैं वो इस बारे में ज़रूर बात करती हैं कि ह्यूमन पॉपुलेशन डिस्ट्रॉय हो जाएगी एंड यू हैव टू रीस्टार्ट एंड यू हैव दोज स्टोरीज आदम ईव मनु श्रद्धा एवरीवेयर एंड इट लुक्स लाइक इट डिड हैपन. We lost a lot of them, and 1280 number was left, And from there, the population came back. Although there is a counter to that, some people said that this happened only in Africa, and it didn't happen, say, in Europe and other places. So the population which was influenced was mainly Africa. How does this information come about? Is population genetics. Okay? Population genetics goes through these models. How the gene flow is happening. So you have to decide what kind of model you are dealing with. And once you do that, then you try to fit your data into that. Finally, you have a lot of markers for population genetics which have been listed here. And some other day I will try to show it to you. Only some important, interesting stories. This is a very common story. This is Drosophila. Drosophila is a fruit fly. The fruit fly has to feed on fruits. Now fruits can be in a temperate region and can be in a tropical region. In tropical region, fruits tend to ferment, and that results in alcohol. So Drosophila, when worked around the world, you see these blue one, that's an allele which provides you resistance against alcohol. So Drosophila in warmer regions has more resistance to alcohol than the Drosophila in the temperate regions. That's one very classical example of population genetics. This is what we did. It's in Nymphia, so microsatellites. What we are seeing here is no variation. So this plant had no variation, although the other species, which is Nosheli, had a lot of variation. So what we figured out later on, that Micrantha doesn't have sexual reproduction. It only multiplies by vegetative reproduction. And Nosheli has sexual reproduction, fully functional. So you find variation in this and not in that. This is Primula. In Primula, we did SNPs. This is only to show you that we did it based on both the microsatellite and SNP. And microsatellite showed us 3 population, which increased in SNP to something like 5 to 6 population. The resolution increased, but microsatellite data was still clean enough to give us enough indications. So that's why we feel that SNP will pick up slowly and microsatellite will be around for a while. So future of population genetics is of course SNP, and microsatellite is going to stay around. And that's all.
Thank you so much, sir, for your wise words and the incredible knowledge that you have shared with us. May I now request our convener, Dr. Savita ma'am, to present the vote of thanks.
Good afternoon, everyone. Thank you is such a prayer that cannot be seen or touched. It must be felt by heart. I, Dr. Savita, feel honored and privileged to get this opportunity to propose the vote of thanks. First of all, I am grateful to Professor Shailendra Goel for providing us his words of wisdom and sharing his time with us. Thank you, sir. I immensely grateful to our principal ma'am, Professor Rama ma'am, for her support and kind motivation. A big round of applause and my special thanks to Vijayarani ma'am for her constant motivation, guidance and support. And I am very much thankful to all faculty colleagues and non-teaching staff of our department who always stand by us and motivate us. I am also grateful to our team of office bearers of Botanic Society. Without their tireless efforts, the success of this event was not successful. A big round of applause for all of them. Last but not least, very warm gratitude to all the members of non-teaching staff of college for providing all the facilities to conduct this event successfully. Once again, I thank you all for your kind attention. Thank you very much.